everyone, I'm Umayangi. I'm from Kurunagala. I'm also joining to the Abhiman Sir science class. So today I'm going to explain the lesson Chemical Basis of Life. The bodies of all organisms are composed of variety of chemical compounds. These chemical compounds are formed by the bonding of naturally existing elements in different ways. There are only about 25 elements in the living body out of the 92 elements present in the nature. The most common four elements found in the living body are carbon, hydrogen, oxygen and the nitrogen. The chemical compounds that build up the living matter can be divided into two categories as organic compounds and the inorganic compounds. Organic compounds in the sense the things which contain carbon, and the inorganic compounds means the things which do not contain the carbon. However, carbon dioxide, carbon monoxide, carbonate and the bicarbonate are the inorganic compounds which contain carbon. And the organic compounds that build up the living matter, they are commonly known as the biomolecules. The main four types of biomolecules are carbohydrate, proteins, lipids and the nucleic acid. When we get the carbohydrate, this is the most abundant organic compound on the earth. The structural unit of the carbohydrate is monosaccharide. Then the common molecular formula of carbohydrate is CxH2OY, and the elemental composition of carbohydrate is carbon, hydrogen, oxygen. And when we get the carbohydrate, hydrogen and the oxygen are combined in the 2:1 ratio. As I told you, carbohydrate can be classified into three types as monosaccharide, disaccharide and the polysaccharide. Monosaccharide is the structural unit of the carbohydrate, and that is generally sweet and the water soluble molecules. Examples for monosaccharides are glucose, fructose and the galactose. When we get the glucose, glucose is something somewhat important, and the galactose, they are found inside the dairy products, and fructose, it is the sweetest sugar. When we get the disaccharide, two monosaccharides join to form the disaccharides. During this process, a water molecule is released, and in the meanwhile, we can get that monosaccharides by hydrolyzing the result relevant disaccharide. It means monosaccharide plus monosaccharide, it forms the polysaccharide plus H2O. So when we get the examples for the polysaccharides, they are maltose, sucrose and the lactose. When we get the polysaccharides, polymerization of the large number of monosaccharides join to form a polysaccharide. Generally, the hydrolysis of polysaccharide also results relevant monosaccharides. Examples for the polysaccharides are starch, cellulose and the glycogen. Starch means that it is not digested starch means the type of carbohydrate that stores in the plant is starch, and the cellulose means it is not digested in the human digestive system, but it helps to avoid the constipation. And when we get the glycogen, the type of carbohydrate that stores in the animal body is the glycogen. When we get the significances of carbohydrate as an energy source to make structural components, and as a constituent of the nucleic acid, and as the storage compound, as a structural component in plant cell wall. So when we get the protein, the second type of biomolecule, protein is an essential constituent in all living cells. When we get the protein, it consists of carbon, hydrogen, oxygen, nitrogen. Sometimes sulfur can also be present in the protein. So 17% of the mature human body is composed of proteins. The structural unit of the protein is amino acid. When we get the amino acid, the simplest amino acid is glycine. There hydrogen is present as the R group. When we get the protein, there is a special term as the essential amino acids. Essential amino acids means some amino acids cannot be synthesized within the body, therefore they have to be taken to the body from the outside with food. So therefore they are known as the essential amino acids. When we get the significances of proteins, they are as an energy source to make structural components as enzymes, as hormones and as antibodies. Then enzymes, special proteins the special proteins that are produced within the organism to increase the rate of biochemical reactions, they are known as the enzymes. When we get the example for the enzymes, to convert sucrose into glucose, sucrose has to be heated with a dilute acid. But the same process is done within the digestive system at a very low temperature. So then we are going to discuss about the lipids. Fat and oil that belongs to this lipid group. Lipids which are solid at the room temperature, they are called the fats, and the liquids, they are called the oils. Similar to the carbohydrates, lipids also contain carbon, hydrogen, oxygen. But compared to the carbohydrate, the level of oxygen is less than the carbohydrate in the lipids. When we get the significances of lipids as an energy source to form different structural components, and for conservation of water to maintain the body temperature, and to protect internal body organ to synthesize some hormones. When we get the lipids, fatty acids plus glycerol, it forms the lipids plus H2O. When we get the fatty acids, we can be classified into two groups as saturated fatty acids and the unsaturated fatty acids. Saturated in the sense fatty acids which contain only single bonds within the carbon atoms, and the unsaturated means fatty acids which contain double bonds with the carbon atoms. Nucleic acid. Nucleic acid is the most important biomolecule out of the biomolecules because of their because of its influence to the when we get the genetical aspect. Nucleic acid is one of the most important thing. So it also contains carbon, hydrogen, oxygen, nitrogen and phosphorus. When we get the structural unit of the nucleic acid, that is the nucleotide is the structural unit. When we get there are two types of nucleic acids as DNA and the RNA. DNA means deoxyribonucleic acid, RNA means ribonucleic acid. The structural unit of the DNA is deoxyribonucleotide, and DNA transfers genetical characteristics from generation to generation. And when we get the RNA is important in protein synthesis process, and RNA, the structural unit of the RNA is ribonucleotide. And when we get the significances of RNA, the significances of nucleic acid, right? The significances of nucleic acid is important in storage of genetical information of organisms, important in transferring of genetical characteristics from generation to generation, and important RNA is important in storing the genetical information of some viruses. And the variations occur in the DNA due to the mutations are important in evolution, and important in controlling all cellular activities in a cell. The information to control the cellular activities in a cell is stored in a DNA, and important in protein synthesis process. Then we are going to discuss about the water. The highest proportion of the body mass of all organisms are composed of water. When we get the water, specific properties of water, a good coolant, a good coolant and a good solvent, and high cohesive and the adhesive force is there, and the differential expansion in the freezing are the significance properties of the water. So when we get the minerals are important as a nutrient constituent to maintain the life process of organisms. Minerals they are absorbed as trace or macro elements into the body. And also when we discuss about the vitamins, they are also organic compounds important in biochemical reactions. Vitamins can be classified into two groups as water soluble and the fat soluble. Vitamin B and C are the water soluble, and the KADE are fat soluble vitamins that we can find. And when we get the test, first one we have to discuss is the starch test. In the starch test, what we have to do is we have to obtain some what we call them a solution of a starch, right? We have to get a solution of a starch, and then you have to put some iodine into that. Then you can observe the purplish blue color appears. Then test for glucose. First of all, you have to get a solution of glucose is obtained, and then you have to put the Benedict solution into that. Benedict, you know that it is in the red color, so Benedict is added to that glucose solution. Then you have to immerse it in a water bath, and you have to heat it well with the water, so you can observe the color change as blue, green, yellow, orange, brick red precipitate. So when we get the test for sucrose in the sense one of the most important thing, it is present in the what we call in the white and brown sugar. Sucrose is there. So a sucrose solution is obtained into a test tube. Then you have to put the Benedict solution, and you have to immerse it in water bath, and you have to heat it well. Then when we heat for the first time, you will not observe, you cannot observe the color change there. So you have to get another freshly prepared sucrose solution, and you have to add the few drops of H2SO4 dilute sulfuric acid into it. Then you have to again put a Benedict solution to that. Then you can observe the color change as blue, green, yellow, orange, brick red precipitate. When we get the test for protein, it means the biurate test. What you have to do is a solution made by grinding dal or an egg white is obtained into a test tube. An extra amount of sodium hydroxide and a few drops of copper sulfate CuSO4 is added into that. Then you can observe the solution, it turns to purple color. So then the test to identify lipids. Sudan 3 test is conducted to identify the lipids. So some amount of gingerly oil or coconut oil is added into a test tube. Then Sudan 3 reagent is added into that. You can observe the appearance of the red blood globules. So I think I did well. So this is my explanation about the first lesson, chemical basis of life. Thank you.